Good afternoon, ladies and gentlemen. Welcome to the distinguished lecture series hosted by our State Key Laboratory of Marine Pollution. My name is Chen Chong, and I'm a postdoc at SKMP. It's my great honor to be your MC for today's lecture. So now, may I extend an invitation to our SKMP Director, Professor Kenneth Liang, to deliver the opening remark for today's event. Let's welcome Professor Liang.
So on behalf of City University of Hong Kong and also our State Key Laboratory of Marine Pollution, we welcome all of you, especially those from online. This talk is in joint venture with the UNESCO. It's called RTRC, Research and Training Center for the Western Pacific region, with 22 country members in the region. We offer them an opportunities for training. If we think the speaker is good enough. No, I'm joking. Today, we are really glad to have Professor Daniel Schlenk, our best friend, to come back and share his crazy idea. I will explain to you why he wants to talk about why animals still surviving, even though being challenged with all kinds of pollutants. So Professor Daniel Schlenk is a distinguished professor of aquatic ecotoxicology and environmental toxicology at the University of California, Riverside. I was unlucky that I applied to be his postdoc many years ago, and then at the end he rejected me. So Dan published widely with more than 400 peer-reviewed publications on the effect of chemical contaminants in wildlife and humans. He has been doing so well. He was elected as a fellow of American Association of the Advancement of Science. You know, AAAS, actually, they published the famous journal Science. So he's a similar level, fellow of AAAS, as well as fellow of CTAC, the Society of Environmental Toxicology and Chemistry. He has served on many important advisory committee and chair for the US EPA on the Chemical Safety Advisory Committee, as well as a scientific advisory panel for US EPA. He's currently the executive and associate editor for environmental science and technology, as well as ES&T letters. He was one of the chief editor for aquatic toxicology and has been serving on many journals editorial board as well. I think today he is going to give a very interesting talk. As you may know, when you're doing toxicity testing, sometimes you work on very toxic compound, but turn out the mortality is 0 after 4 days. This happens all the time. Then the question is, how can aquatic organism to fight against the odd situation is something we really overlook over the years. Because if we understand the cure, actually, it will help us to manage the chemicals better. Okay, so please join me to welcome Dan.
Thanks, Kenny. Actually, Kenny turned me down. It wasn't me turning Kenny down. So just make sure we get that straight. So thank you very much for coming today, and those of you online for tuning in. Really appreciate the opportunity to chat with you a little bit about some work that you may not know that I have done in the past. As Kenny said, it's a little bit different than some of the other stuff that I've done. As you mentioned, my focus area has been primarily ecotoxicology. And you know, as you get older, you start thinking about things like, why did I do this? And as students, it's something I think you need to ask yourself pretty regularly is, why am I doing why I'm doing this? What are the things that are driving me to do this? And in science, it's really important. I think you have to have a, you know, a passionate heart to deal with things, because sometimes things don't work so well. And so you have to fall back on that passion to figure out how to move forward. So, so this is a little bit of a historical sort of venture for me. So this sort of began actually for me when I was about 13 or 14 years old, because I wanted to become Jacques Cousteau. If you know who Jacques Cousteau is, I don't know if maybe the younger people in the audience don't know. He's the person who discovered the self-contained breathing apparatus that allows us to go scuba diving. And when I was a young child that used to make these programs on TV, and it was, you know, diving in these amazing places and seeing all these amazing animals, and it was like, wow, I want to do that. And so I was fortunate enough to live in Southern California on the coast, and because of that, I was able to go diving quite a bit and see a lot of different animals. And then one day, a friend of mine, we just happened to be in an area of the Los Angeles Harbor. And if you know anything about Los Angeles Harbor, it's like Hong Kong Harbor. It's not very clean. And diving through the harbor, I looked and it's like, well, I didn't expect anything to be alive in there. And going around and looking through these different locations, you can see, this is a picture that a friend of mine took from the harbor. And you can actually see there's animals that live in Los Angeles Harbor. Very contaminated location. You can see there's a sea star here that's present here. And you can also see holes from polychaete worms that are basically also living in this location. And they seem to be just doing just fine in a location that's really, really contaminated. So my question was, how in the world are these things actually surviving? And how do animals actually survive in these situations? And, you know, situations can vary all over the world. So this is Los Angeles Harbor, but, you know, animals can survive in very, you know, crazy environments as well. You know, some of these animals, you know, we just don't understand how they can survive the next years. You know, it's just crazy. So we really need to sort of understand how these animals move on. So this is why I actually got into toxicology, was to ask this question. So if you ever took one of my classes, this would be the final exam question that was always present, is why doesn't exposure equal toxicity? Because we're exposed to chemicals all the time, and animals in the environment are exposed all the time. But in order to actually have toxicity, you actually have to have absorption, number one, and you have to have distribution to a target. Now that target can be the brain, it could be the heart, it could be the muscle, it could be anything, but you still have to have movement and exposure at that site in order to have toxicity. Okay, now, how do we prevent toxicity? Well, there's a number of different ways that we can prevent toxicity. One of those is to take that chemical that is usually very lipophilic and very hydrophobic and change it into something that is water soluble and easily eliminated. Right? That's one way we can do it. The other way we can do it is by storing it and putting it away someplace so that it doesn't get to the target, and maybe moving that storage to maybe a kidney or some other organ that allows that chemical to get out. Plasma binding proteins, for example, serve that purpose. Bone serves that purpose for metals. If you're a bivalve, sometimes the shell itself actually serves that purpose as a detoxification strategy. So, but the downside of that is that by changing the chemistry of the chemical to try to get rid of it, oftentimes we make that chemical more toxic. So this is again, was one of the reasons I got into toxicology. I found it fascinating that you could take a chemical and put one atom on that chemical and kill an organism. And by one atom, that atom can be oxygen. Just by putting one atom of oxygen, you can kill an organism. So this is one of the things that sort of drove me. And, you know, I've also kind of got interested in aquatic organisms. So we try to apply this to aquatic systems. Now, the process that I was really interested in was this biotransformation process. And if you look at this cartoon, this was actually done by a colleague of mine in 1985. This is when I was in graduate school. We were both in graduate school together. He was a little bit more artistic than I was, but he put this cartoon together to demonstrate what happens at the endoplasmic reticulum of the cell. This is an organelle that is in all cells, and within this organelle are the enzymes that are responsible for this biotransformation to take place. And one of the most studied enzymes within the endoplasmic reticulum is this big thing right here. This is the cytochrome P450 monooxygenase system. Now, enzymes are great because they always tell you what they do. So a monooxygenase is going to take one atom of oxygen, and what it does is it takes a nonpolar lipophilic compound, in this particular case, a polyaromatic hydrocarbon that comes from oil and combustion byproducts, and actually makes that compound hydrophilic and more water soluble. This process is called phase 1 biotransformation. In so doing, it oftentimes makes this chemical more reactive. So the cell has evolved other systems, such as the glutathione transferase, which is present here, which transfers this tripeptide called glutathione onto this molecule, making it again more water soluble, bigger, and more readily eliminated. That's a phase 2 biotransformation process. So again, through these phase 1 and phase 2 reactions, cells can detoxify organic chemicals and make them less toxic. But in some cases, again, they can make them more toxic by putting oxygens in the wrong place at the wrong time. So what's this got to do with the talk that I'm giving today? Well, when I started graduate school, I actually really wanted to become a marine biologist, as I mentioned before. So I wanted to go diving in really cool places and kind of, you know, collect animals and do a lot of work on them. So the very first organism that I worked on was this big thing that looks like a rock. It's actually an animal. It's called a chitin. It's a very old animal in terms of evolutionary age. And you can actually tell what it is. It looks like a big rock that's present here, but underneath that skin are 8 plates that are present. And it basically is a gastropod snail. And what's really unique about this snail is that it's about this big, right? So they're very easy to collect, and they have a very big mouth. Okay, now why is that important? Because if you want to expose the animal, you can actually feed through the mouth with a gavage needle like you use for rats and actually expose it to anything you want. The other unique feature about this animal was that it also had a very, very large digestive gland, almost 20 grams. Now, again, to study the enzymes for this biotransformation, you need a lot of biomass. So using this biomass, we could actually use one animal and get a lot of tissue sample to look at how the animal metabolizes or biotransforms these different organisms. Now, again, the title of the talk is Why Don't They Die? Well, this is an animal that I was very fascinated with because it feeds on this particular algae called Odenthalia flacosa. It makes up about 25% of its diet. And basically, it only exists as about 10% of its available food. Now, why is that significant? And why is that significant in terms of this particular talk? Well, when we started looking at sort of the enzymes of biotransformation that were present, we found very significant levels of those phase 1 cytochrome P450 in the intestine and the digestive gland of this animal. But one of the things that jumped out of us was this enzyme called glutathione transferase. You can look at the units here. These are micromoles per minute per milligram. The human liver, you and I, only have nanomole per minute per milligram levels of this enzyme. So the question we started asking was, why does this animal have so much of this detoxification enzyme? Well, it turns out, if we look at that Odenthalia algal species, it actually keeps a lot of other animals from eating it. We call this chemical defense. And the way that it does that is by making this chemical called lanosol. Now, those of you that look at environmental contaminants might look at this and see, wow, that looks kind of familiar. And the reason why is because if you take this compound and you mirror image it and dimerize it, what you are creating is a PBDE. You're actually making a polybrominated diphenyl ether. Has anybody ever heard of those compounds before? Yeah, they're flame retardants, right? That were used forever. In fact, you can probably look on your chair right now, and I bet some of those chairs actually still have some of those compounds that are present. Now, most of those have been replaced by other brominated flame retardants that are also present, but the idea is this is a natural product that occurs within the diet of this animal. And what's interesting about the structure of this compound is that it lends itself, because of these halogens, to being very good substrates for glutathione transferase. So we stated the hypothesis that maybe the reason why these animals have such high GST levels and P450 levels is this may be how they are detoxifying this product. So we took this chemical. I had a very good friend at the University of Mississippi who actually synthesized this compound for me, and we were able to feed it, again, gavage through this mouth of these animals and actually look to see what happens. And what was interesting was that we could actually see induction of a P450 that we see in us called CYP3A. Now, the exact enzyme in this particular organism is not 3A, but it's 3A-like because it carries out the same enzymatic activities that the 3A does in humans. And you can see it's actually increasing after exposure to this particular compound. Again, giving us insights that maybe this is how this compound is actually detoxified. The other really interesting aspect of this was when we looked at its exposure to glutathione transferases, we actually found a really unique situation that took place. What we found was that when we co-expose the enzyme with lanosol at different concentrations, you actually can see that the compound is actually non-competitively inhibiting glutathione transferase activity. What does that mean? That means in the active site of the enzyme, where the enzyme is actually working on a substrate, in this particular case, it's called CDMB, lanosol is coming in and blocking the active site so that the chemical cannot be metabolized. Now, we would think that this is probably because this compound is metabolized, but that's not always the case. What it does indicate is that it's entering the active site of this enzyme, and maybe this may be how the animal is detoxifying its diet, and thus maybe being protected by other human-based contaminants that it may come in contact with. So if we look at just sort of a summary of this particular sort of study, we can see that we do see a P450-like enzyme that's present in cryptokinin, that it's induced by lanosol, and that lanosol, again, non-competitively inhibits this phase 2 enzyme called glutathione transferase. Now, a lot of this work was done in the Northwest, and when I moved back to California, I thought, well, let's follow up this study a little bit more and look at animals that have similar feeding strategies in Southern California. So we looked at another chitin species called Catharina, which again has a very similar feeding strategy as the gumboot chitin, which I talked about before. It feeds on filamentous red and brown algae and is considered a generalist, meaning it eats a little bit of everything, just like the big chitin that we had in Oregon where I had studied earlier. The other animal that fits the same niche ecologically is something maybe you are familiar with in China. Abalone. They're very, very yummy to eat, and they're cultured and actually readily available. So we wanted to compare this particular chitin, and here you can actually see the 8 plates along the base of the spine of the snail, and compare it to this species, which basically avoids lanosol-containing algae and feeds mostly on kelps, on brown algae. So we would, again, hypothesize that maybe because of the different feeding strategies, they would have different detoxification mechanisms. And sure enough, if you look at how these different species compare to one another, you can see the P450 mediated reactions are very, very different between the two in terms of how they are inducible. So with chitins, with the brown chitin that what I showed you before, we saw induction of those CYP activities. Here, we do not see induction. But what's really interesting is if we look at GST, we actually do see almost a twofold induction of the chitin, but not the abalone. So it seems to be the chitins have evolved GSTs to deal with these dietary contaminants, if you want to call them contaminants, to their diet. So if we again look at sort of a quick conclusion here, we can see that definitely different CYP expression between chitins, whether we're looking up in the northwest of Oregon versus Southern California. We do see reduced catalytic activities, but induced catalytic activities in the Oregonian species. Lower basic levels of CYP activity were seen in Southern California, and a little bit lower than what we see in abalone. Lower levels in the Southern California species, but still inducible by lanosol, which is something that they rarely see in their diets. Now, again, this type of work was done in the Pacific Ocean, which again is a little bit chilly to go diving in to actually collect these samples. So I started thinking, where's a really cool place that I could go that's actually a little bit warmer? Well, turns out that I decided in 2003 to do a sabbatical in Australia. And in Australia, you have, and in Southeast Asia in general, and the South Pacific, we have this particular group of fish called the butterfly fish. Now, does anybody know what is unique about butterflies? That they have very unique coloration. Does anybody know why they have the coloration like that? It's actually a warning. It basically says, don't eat me because I am very toxic. It's an ecological strategy to keep things from eating them, okay? Butterfly fish do the same thing. They have this big warning level, as do other tropical fish species. Why? Because they feed on things that have chemical defenses, and they take that chemical defense and they incorporate it usually into their gonadal tissue to keep things from eating their gonads. And again, it's another warning strategy to say, don't eat me. What's unique about butterfly fish, though, is that there's about 65 different species of them, right? And they all have different feeding strategies. Same genus, different feeding strategies. Now, the other thing about them is they all have cytochrome P450 as well. And a lot of work has been done by a guy named Nick Brolick in Valerie Paul's lab in Guam that actually characterized P450 in a lot of these different species and indicated that there may be an association between what they eat and these P450s that are present. So what we wanted to do was sort of follow this up. And the way we followed it up was by looking at a very specific group of compounds called episimulepulides, which are present in this coral called Sinularia maxima. Now, Cineraria maxima, I think I had the picture of it that's present here. This is a soft coral that's present in the South Pacific, Australia, Guam, and certain butterfly fish species will eat this, but other species will not. So what we wanted to do was take extracts of this soft coral and just like we did with the chitin, gavage them within butterfly fish and see whether or not their detoxification strategies could be changed. So this was a lot of work that was done primarily by myself and a graduate assistant on the Great Barrier Reef. And I thought this will be really, really fun, right? Get to dive in this amazing place. It was probably the hardest thing I've ever done in my entire life. Trying to catch these things in the ocean, bring them up alive to take to the laboratory so that you can stick a needle down their throat and inject this extract to see what happens. Needless to say, we maybe got about 50% survival rate out of most of our animals that could actually do this. So it was very, very difficult, but we wanted to give it a try and see what happened. So the idea here was to look at the sequences of these animals for P450 to see if they corresponded to their feeding strategy, and then actually see what this extract did to these P450s that were present. And then use this isolated natural product from, and I'll get to this a little bit later, of 5-epicinyleptylide to see what it actually did to these animals. So the first experiment, again, was to sort of look at the sequence homologies of these different species and see if they grouped into different sort of niches. And what we see is that if we look at cytochrome P450 1A, which is probably the most studied cytochrome P450 in fish, you can see that it does actually group within different feeding strategies. We can see that some of these animals are exclusively coral polyp feeders. Now, again, why is this important? Have you ever thought about looking, when you look at a coral reef, how beautiful it is? Actually, these seats remind me a lot of the coral reefs because when you look at them, nothing is on them. If I took a boat or a wooden stick and I stuck it in a coral reef, within 24 hours, I would have slime and algal buildup on that stick within 24 hours. If I left it there for a week, I'd have barnacles on it. I would have all kinds of fouling organisms. Do you ever see that on corals? Do you ever see it on sponges? Do you ever see it on any of these animals? You don't. Why? Because they're chemically defended. They actually make anti-fouling compounds to keep them free and clear so that they can feed and live their life histories. So again, looking at feeding strategies, if we see things that exclusively eat coral polyps, we would think maybe they have a different defense mechanism to actually consume these organisms. If we look at generalists, which are present up here, none of these animals, particularly this C. auriga, eats everything, right? It eats polychaetes, anemones, algae, as well as coral. I'm going to talk a lot about this particular species, auriga, Chaetodon auriga, a little bit later. One of these actually comes out and does soft and hard coral polyps, again, in Australia, and actually has its own sequence variability. If we look at the rest of these species that we looked at, these again seem to be more specific towards coral polyps, but also now and then will actually eat benthic invertebrates. One of the ones I want to highlight here is this particular species, Punctophacialis, and then these two, Kleinii and Unimaculatus, which we're going to talk about a little bit later. So again, this is sequences showing grouping for this particular P450 CYP1A. So what happens when we actually take these and basically force feed them with a Cineraria extract and then look to see what happens with expression? So again, this is sort of repeating the experiment that we did with the chitins, except with a extract from a soft coral. And what you see, particularly with the CYP1A, again, the groupings of CYP1A, you see differences in expression, but you don't see really any induction. What you do see, interesting though, is differences in expression between species. And again, we see Punctophaciatus being a little bit less than some of the other different species, particularly the generalists that we see with Auriga. And again, the semi-generalists, if you want to call Kleinii that, between this species and this species, we see a reduction that's present, but no real induction. So we thought, well, let's look at some of the other P450s that are present, not just 1A. Let's look at some of the constitutive P450s like the CYP2 families. And looking at this species differences in terms of sequence, we see that each individual animal has their own CYP2 based sequence. Very, very unique for each of those animals. So it's very similar to humans. If you looked at my CYP2 profile, my CYP2 profile is different from yours. And it's very individualized, which makes drug therapy a very unique sort of process. Sometimes people respond differently to drug toxicities because they have different CYPs associated with them. And such is the case, it seems, with butterfly fish. So again, if we look at these CYPs, we actually see a completely different pattern, right? In this particular case, we see that puncta fasciatus, instead of seeing no change, we actually see an induction that's present. We still see these reductions that are present, but this induction, again, in this soft coral polyp specialist, seem to be fairly unique. So if we're looking at conclusions here, we'd see that obligate coral feeders group with sequences for 1A, but there's no group for these 2Ns that are present. And what's really unique is we're seeing induction of the 2Ns, but not the 1As that are present. And a general repression of CYP1A in the other species following the soft coral. So what this means is those species that eat soft corals seem to be getting induced, even though they have a lower level of overall CYP that's present. So, as I mentioned, collecting these animals was really, really difficult. Very difficult. One day at a scientific conference, I ran into a guy, his name was Gary Ostrander, who actually was the dean of the medical school at the University of Hawaii. Turned out he used to do a lot of work with corals. And he was very fascinated, and because he had become an administrator, he really wanted to keep doing research in corals. And so I told him about some of the work we were doing in Australia, and he said, wow, wouldn't it be interesting to compare Hawaiian species of butterfly fish and Australian species of butterfly fish? And the reason being is in Australia, we have soft corals and we have hard corals. In Hawaii, it's almost exclusively hard corals. So Sinularia, that yellow coral I showed you before that has epicinuleptolide, does not occur in Hawaii. But the same species that eat that do. So wouldn't it be interesting to compare the same species that have the same feeding strategies, but different targets to determine what their detoxification pattern would be? So I had a student, Eileen Maldonado, who actually lived in Hawaii, which was very nice. So she could get her PhD degree at UCR and live in Hawaii and actually conduct these studies. Gary Ostrander, because he was dean of the medical school, actually controlled Waikiki Aquarium. So I could actually go out, get a guy to sample every species of fish I wanted for free and bring back 30 to 50 individuals that were totally healthy and do these experiments. It was fantastic. It was so much easier. All I had to do was just show up and actually gavage the fish and then look to see what their P450 levels were like. So it was a real interesting and fun relationship that we had with Gary, and we still work together on a number of different things. But so these are the different species that we looked at. As you recall, Kleinii was one of the polyp feeders that was present. And in the South Pacific in Australia, it's an obligate soft coral feeder, feeding on this soft coral called Lobophytum. In Hawaii, because there are no soft corals, it's a sometimes, that's what facultative means, a sometimes hard coral feeder and predominantly a plankton feeder. Okay, same genus, same genetics, same species, different feeding strategy. Okay? If we look at this particular species, Chaetodon lunatus, it's an obligate hard coral feeder and feeds on Acropora. In Hawaii, again, because it has hard corals, but not Acropora, it feeds primarily on Porites, but it's still a hard coral specific feeder. Auriga, which we talked about before, is a generalist in both places. So it eats everything, everywhere. Unimaculatus was one of those, if you recall, was an obligate soft coral feeder in Australia and Guam, but in Hawaii is a hard coral feeder. Now, what's unique here, and very, very important to point out, is that it feeds on Sinularia. Okay, it feeds on that yellow species of toxic soft coral that's present. So I go back to my little cartoon again, just to remind you of the different enzymes that we're after here. And again, we're after this cytochrome P450 enzyme that's there to make things less toxic in our hypothesis. So what do? We measure not only that particular enzyme, but we measured, again, these phase 2 enzymes like glutathione transferase, epoxide hydrolase, and other phase 2 enzymes, which are not in this cartoon, but also play a role in detoxification. So these are all the different activities and enzymes that we looked at and compared them between Australia and Hawaii. Now, again, due to time, I'm just going to focus on the 2 species that I thought were the most interesting. And that is this generalist, that is a generalist in Hawaii and in Australia. And what you see here, the black bars are the Hawaiian responses, the white bars are the Australian responses. What we see, particularly with cytochrome P450 1A, is that the basal levels of expression in Hawaii are significantly higher than what we see in Australia. However, if we look at the CYP2 and the CYP3 sort of isoforms of P450, we actually see there's not a whole lot of difference between the two, maybe a little bit higher in Australia with the generalist. However, when we look at CYP2 protein, this is again looking at messenger RNA. If we look at the protein is higher and the activities are higher in terms of the generalists in Australia. If we look at some of the other enzymes, we generally again see greater activity in Australia than we do in Hawaii that's present there. So does that mean there's something unique in Hawaii that's different than Australia? Well, we think so. And in fact, if we look at Unimaculatus, this is the one, again, that's the soft coral feeder in Australia, hard coral feeder in Hawaii. Here's our 2N that was inducible again in Australia species. It's inducible in this species as well after or looking at Australia versus Hawaii. So again, generally speaking, it looks like the Australian species with similar or somewhat different feeding strategies tend to have higher detoxifications. The only difference was with Auriga, which had higher CYP1A. The question would be, why would Auriga have higher CYP1A? Well, again, if you know anything about CYP1A, it's inducible by pollution. So we think that Hawaii, the animals that were collected in Hawaii, at least for Auriga, might have undergone a little bit of pollution. And if you know anything about Hawaii, there is this really, really big harbor there called Pearl Harbor that actually has a lot of sunken ships that are present that are actually leaking out oil that would actually could induce this CYP1A. Again, hypothesis, but it does seem to be somewhat consistent. But the bottom line is we see generally higher detoxification strategies in Australian species than we do in Hawaiian species that's present there. So let's go back to this Sinularia sort of phenomenon that's present. Why is Sinularia not fed upon by different species? Well, again, we think it's because of this compound called 5-epicinuleptylide. So my colleague at the University of Mississippi, in collaboration with Valerie Paul, actually looked at these different species in Australia and actually looked at their feeding strategies to determine whether or not this compound could deter the feeding of their natural diet. So what he did, he took agar blocks and took 5-epicenoleptylide and put it in the agar block and looked to see whether or not it could deter feeding. And what you see is that with Auriga, the generalist, it does not like 5-epicenoleptylide. It'll eat it, but it really doesn't prefer it. However, when we look at Unimacculatus and Melionotus, you can actually see that it actually doesn't care whether it has 5-epicenoleptylide in it or not. So again, this would indicate to us that these animals are able to eat this and deal with this compound somehow. So we wanted to follow this up. So because Mark had isolated the compound, the previous studies that we looked at the extracts. What happens when we take that compound alone out of the extract and see what happens to it? So in this particular experiment, we took, again, our Auriga generalis feeder from Hawaii. We took Kleinii, again, a facultative coral feeder in Hawaii. And we took Multicentus, which is an obligate hard coral feeder from Hawaii. And then here's Unimacculatus, our obligate hard coral feeder in Hawaii, but Sinularia eating feeder in Australia. Okay? So again, these are all Hawaiian fish, never seen this yellow soft coral before in their lives, had never seen 5-epicenoleptylide in their lives before, right? What happens when we treat them with this compound? So, interesting thing happens. If we treat these animals with this compound, the soft coral specialists from Guam, or from Guam in Australia, 100% survival. The generalists, C. Auriga and C. Kleinii, we actually see a dose-dependent survival of response. And when we see the hard coral specialist, that's, again, never seen this compound, 100% mortality. So this compound is toxic. It kills fish. But yet, it kills the fish that have never seen it before. But this particular species had never seen it before either, except in Australia. So how in the world is this animal surviving? Why doesn't it die? So we started looking again at the cytochrome P450 levels. In this particular case, we looked at CYP3A. CYP3A was very, very high in natural levels in terms of basal levels when we compared the three different species. When we looked at the catalytic activity of that enzyme, again, fairly similar responses between species, with Auriga having the highest. And again, Auriga generally had the most protection. But what about Unimacculatus? How come that actually survived? Well, if we actually look at the treatment levels of those different species, we can see that if we, again, gavage these with a low and a high dose of this compound, what happens is Auriga, nothing, Kleinii, nothing, Multicentus, nothing. Even though Auriga has higher levels, nothing really happens. But look what happens to Unimacculatus. Again, this is a species that has never seen this compound before. Okay? Genetically it has, and genetically it seems to have this capability of detoxifying this compound, and it's inducible. It actually is able to induce this defense. And we see this not only, again, in protein expression, but also in catalytic activity with the animal as well. So these are catalytic activities associated with CYP3A, and they also go up with treatment with 5-epicenoleptylide. If we look at the CYP2N that we looked at before, again in Australia, we see a similar pattern. Again, a little bit of induction that's present here, nothing statistically significant. In fact, actually a somewhat reduced catalytic activity that's present. But with Unimacculatus, again, this induction of the CYP2 form that's also present that seems to be going up. So, are these CYPs responsible for metabolizing this compound? Are the CYPs actually present for detoxifying this chemical? So, again, the great thing about science is you get to collaborate with people from all over the world. So, in collaboration with a guy named Jed Goldstone at the Woods Hole Oceanographic Institute, who's looked at the phylogeny of P450s, he took the CYP3A model and actually integrated 5-epicinyleptolide into the active site and was able to predict metabolites that we would see. And one of the metabolites that we should see is this epoxide. Again, a monooxygenase reaction to an epoxide, which would then open up to a diol or actually metabolites of other locations. But it was this epoxide that we thought would be really interesting to look at. And it was the highest predicted sort of metabolite from the modeling. So whenever you see a molecular model, it generates a prediction. You have to follow that up with a hypothesis. So our hypothesis was that P450 is metabolizing this to the epoxide. So how do we prove that hypothesis? Well, what we do is we have to make that metabolite and look to see whether or not the animal actually generates that metabolite in terms of interactions with P450. So again, my colleague at the University of Mississippi, John Romaldi, actually made the epoxide from 5-epicinyleptolide, which allowed us to actually do quantitative measurements of catalytic activity within these different species. This is the HPLC trait. This is the mass spec that actually shows the confirmation that the compound is indeed the epoxide that we're actually looking at and measuring in these different species. So again, what happens when you compare the different species with the different feeding strategies, looking not only at clearance of 5-epicinyleptolide, but also the epoxide formation? So again, Auriga is the generalist, which again had a dose-dependent survival and had endogenous high levels of CYP3A. What you can see is it clears it pretty readily, right? About 200 picomoles per minute per milligram. And it generates the epoxide. Again, consistent with our hypothesis that the animals that have high CYP3A are detoxifying this compound. If we look at Kleinii, which is again was one of the compounds or one of the species where we saw the dose-dependent protection, it generates the epoxide as well, but in a very lower amount. Again, fairly consistent with the toxicity that we see. Multicentus was the one that died, 100% death rate. And you can see that it does not make the epoxide. 0. Okay? And it has very low levels of CYP3A as well. If we take Unimaculatus, again, this is our superstar fish from the Australia soft coral feeder that is now a hard coral feeder in Hawaii. You can actually see it is the highest clearance of 5-epicinyleptolide and almost and a very high level of epoxide formation. What's very interesting is if you take Unimaculatus and you pre-treat with 5-epicinyleptolide, you can see that it actually significantly makes almost 20 times higher amounts of that epoxide after induction. Again, very consistent with CYP3A. If we actually look at the human CYP3A form, the human CYP3A form even metabolizes this compound, which would indicate that you and I could probably eat this coral and probably survive it. And it's actually consistent with what CYP3A does. CYP3A is what allows you and I to eat broccoli, celery, mushrooms, which, by the way, are all incredibly toxic plants, right? But very good for you. Why? Because they contain other vitamins that we need for our livelihood. But they're very toxic. Why are they toxic? To try to keep insects from eating them. We have evolved these P450s, just like these fish have evolved these P450s, to detoxify their diet so that they can survive. This to me was probably one of the best sort of evidence that indicated CYP3A is basically responsible for epoxide formation. If you look at the actual epoxide formation concentrations and the CYP3A catalytic activity, it's almost a direct correlation. Again, consistent with 3A being the detoxification mechanism that's present, at least in this genus of animals. So again, if we look at conclusions, 5-epicenoleptylide had the greatest toxicity on the hard coral specialists, but the least toxicity on the non-coral feeding generalists and the soft coral specialists, Unimaculatus. Basal levels were highest in C. auriga, but this was inducible when animals were treated with this anti-feeding compound. If we look at the molecular modeling studies, this predicted that CYP3A would convert this to an epoxide, which it does when we actually measured it. There was a correlation between this and the epoxide formation in all species. And again, the bottom line here is treatment with a South Pacific species that had never seen this before. You see metabolism in a different species in Hawaii. So very, very unique sort of relationship that's present. And again, indicates that P450 is really, really important. So again, a lot of this work was done in Hawaii, Australia, very long distances for me to travel from California. I needed to find something that was a little bit closer to California. So fortunately for me, I had a student very interested in another species of sea organisms called sea turtles. Sea turtles are also herbivorous. They have very unique feeding strategies. Green sea turtles primarily are herbivores. They eat seagrasses, right? Loggerheads actually eat a whole bunch of other things, particularly plastics these days, but they actually are trying to eat mostly cnidarians and jellies, right? As well as invertebrates. So again, they're generalist feeders, but the really interesting species of sea turtle that we were very fortunate to deal with were hawksbill sea turtles. Now, what is unique about hawksbill sea turtles? They actually eat sponges. They preferentially eat sponges. And again, sponges, what makes sponges so unique? They are chemically defended. That's why things do not settle on sponges. They make a very unique compound called dibromoperol carboxylic acid, which is present here. Looks very similar to lanosol, doesn't it? Very, very similar to the bromination pattern that's present. Again, Valerie Paul had indicated that this is chemically defensive. They actually will deter feeding from fish species in Guam. Well, what about turtles? How does this turtle eat this sponge and detoxify this compound that's present? So again, looking at the structure of that compound, looking at lanosol, and then thinking about those chitins that we looked at before, one of the enzymes, again, that was really important in that species was glutathione transferase. So what about glutathione transferases in sea turtles? Was there any unique sort of pattern that was present there? And if we look at the different GSTs based upon their catalytic activity, and if we compare the species between so these are all color-coded based upon their feeding strategies. If we look at green sea turtles, which are in green, and hawksbill sea turtles, which are in yellow, you can again see that pi GSTs are actually much higher in hawksbill sea turtles than some of the other GSTs that were present. What's really unique here is that if we look at the same substrate that we looked at in chitins, actually, this was a little bit lower than what we saw. Still micromolar per minute per milligram concentrations, which are again higher than humans, right? Still very, very high levels, but again, didn't really match the feeding strategies. So again, John Romaldi made this compound for us, and we were able to look. We couldn't feed turtles this because this is an endangered species, so you can't go out and feed them and actually kill them and look at their livers. But we could actually incubate them in terms of the GST cytosols that we had collected from the livers of samples. and look to see, again, whether or not they changed activity. And if we look at, again, the pi activities, we really don't see a whole lot of change. We do see, again, that hawksbills had higher levels, but with treatment of this natural product, no real change is present. And in fact, if we look at CD and B, the only real difference that we see is a reduction of activity. So that made us look, well, what about the kinetics? Do we actually see the same type of relationship that we saw with chitins? And sure enough, if we actually look at the kinetic relationship between the dark lines that are present here, you can again see a non-competitive inhibition of glutathione transferase activity that's present here. Again, same affinity constant, meaning it has the same affinity, but lower Km, or sorry, lower Vmax, leading basically to lower turnover, almost half as much turnover when you treat with this, again, anti-feeding compound. So we thought, okay, this is really exciting. Let's look to see whether or not we get metabolism. So my student, Christy Richardson, who did this work, we got radio-labeled S35 glutathione, and we did incubations with DPPC, expecting to see conjugation with glutathione. What we saw nothing. The only thing we saw was glutathione oxidation, which indicated oxidative stress, really, and no metabolism whatsoever. So we started scratching our heads. Well, how can this happen? It's inhibiting glutathione transferase activity. But remember what I told you before, right? Non-competitive inhibition means that it's binding in the active site. It doesn't necessarily mean it's being turned over. And so Christy started looking through the literature, and what we found was that not only are we seeing competitive inhibition that's present here, but this non-competitive inhibition was actually seen in other molecules, particularly bilirubin. Now, bilirubin is actually a breakdown product of hemoglobin. Okay? Hemoglobin this is actually a very toxic compound. In fact, babies, if they are not metabolizing bilirubin, this will actually back up in your blood and turn to jaundice. This is why you get the yellow coloration of jaundice. And it's actually it actually attacks the central nervous system in babies. So consequently, you want to metabolize this very, very readily. And what Christy found was that in mammals, hemoglobin breakdown products, bilirubin, are actually picked up by glutathione S transferases and transported to the bile where they're eliminated. So GST is actually not acting as a biotransformation mechanism, it's actually a storage mechanism for bilirubin. So we decided, well, what happens when we take bilirubin and incubate it with sea turtle GSTs? And what you see is the exact same relationship that we see with DBPC. If I go back, if we go back one slide here, same relationship with DBPC as we see with bilirubin. So what this indicates to us that the defensive mechanism in sea turtles and possibly in chitins with GST is not catalytic turnover or biotransformation, but it may be storage. So overall in turtles, we see again this high GST pi in hawksbills, the sponge-eating species, a non-competitive inhibition by DPPC, but no metabolism, but potentially a storage mechanism. So if we go back to our why doesn't exposure equal toxicity and why don't they die sort of paradigm, what we see is that what happens is not only do we see biotransformation leading to elimination, potentially like we see with butterfly fish, but we may also see storage as a mechanism that moves natural products and anti-feeding compounds to elimination and keeping them from the target as a mechanism of detoxification. So with this, a lot of people to thank. A lot of people collaborate on this project. This has been something that I've been working on since about since 1988. I think I published my first paper on this. Many of you probably weren't even alive then, but it's been something that's been really interesting and something we've been able to continually sort of look at. And hopefully I can interest a few more students along the way to do this in the future. So with that, I'd be happy to take any questions that you might have.
Thank you, Professor Schlenk, for your enlightening presentation. So now we have some time for the Q&A session, and any questions are welcome. So, okay.
I can call you marine biologist.
Yeah, thank you.
Pass. So you name many species that I think most of them didn't know about. I never seen this gigantic chiton. It's really big. I just checked on the internet. It's huge.
Yeah,
Are they tasty?
I don't know. I never tried, never ate it. Didn't eat it.
So my question is, I really enjoy your talk. It seems to me the evolution take place when the organism being exposed to those toxicants material, and then they develop this SIP SIP proteins or enzyme to deal with the situation. Can I say that if, say for human, if we eat many different things, especially those potentially toxic things, we may induce different kinds of metabolism or detoxification. Then over time, we can become more accommodative or resilient to those challenges. But if the chemicals of those toxicants very similar to other pollutants, then we have the ability to fight against the odd. But my question is, can we make a super organism by cloning all kinds of SIP into the fish? Then whether this fish become immortal in some way, I just want to hear from you. And sometimes it explains why Chinese and also Japanese live longer, because we eat many different things.
Yeah, it's very possible. Definitely more vegetable-based diets tend to live longer. That's known, right? If you look at the centenarian populations of Japan and Italy, and even in California, vegetarians tend to live longer because they have the capability of dealing with those compounds. In fact, it's really interesting is if you look at cooked meats, they're very toxic because the P450s that we have actually make those compounds more toxic, which is really a unique sort of comparison. But yeah, in terms of making an AI fish that overwhelms, well, I mean, a really unique situation is some of the fish species in contaminated environments, the fungulus species. I don't know if you're familiar with some of the work that's been done there. Cole Matson and Rich DiGiulio's work with fish that survive in oil contaminated estuaries. What's really unique about those animals is you take an animal that's never been exposed to oil and you put oil in that animal, it kills it. If you take that population of animals from the Elizabeth River and expose them to oil, they're fine, totally fine, and they'll survive and reproduce no problem, right? If you hybridize them and look at both females from that population compared to the other, you can pass that trait on to the next population. So there is precedent for evolution and adaptation through this mechanism. I also point out almost all of this work is all done with insects. Insect resistance is how this is how insects become resistant. They do the exact same thing. They induce an enzyme that detoxifies the pesticide that makes it irrelevant. And that is actually, and that's not even man-made pesticides. These are natural compounds produced in the plants. In fact, May Berenbaum, if you're familiar with her work, it's called the chemical arms race. Plant makes deterrent, insect evolves P450 to eat the deterrent. Plant makes another deterrent, insect evolves another P450 to deterrent. So it's a really common aspect, particularly with invertebrates, that's present, and also in us. You know, I think with us, it just takes a little bit longer. With vertebrates, it takes longer. But definitely it's explains resistance, at least in insects and in also, I think, tolerance and adaptation in other species, for sure.
Okay, thank you. So any other questions? Okay.
Oh, hey, Jimmy.
First, very much, thanks for a lot for your inspiring talk. And my question is related more broadly and about in the long run, can we really map the toxicokinetics and the toxicodynamic variability among species so that to map this kind of aligning with the species sensitivity distribution?
Yeah, I think
there a
so.
way to go? Yeah.
Yeah, I think so. I think it's possible.
Otherwise, we may test the species by species. So.
Sure. I would foresee, for example, using an omic-based strategy that gives you all of the enzymes, right? All of the different aspects and the variants that are present between individual species, and then modeling that based on turnover rates to the pharmacokinetic aspects and looking at clearance rates of different compounds and actually tie that into a phenotype of a species to determine sensitivity or not. I definitely think that's possible. Maybe in our lifetime.
Maybe the large language models can help.
Pardon me?
The large language models.
Yes, definitely with ML. That's what I'm thinking. Yeah.
Speed up this process.
Yep, definitely.
Another question is related to 6-PBD and the 6-PBDQ
Mhm.
molecules. These molecules are relatively strange in terms of the species sensitivity,
Yes.
right? Not quite discrete.
Very,
A
very,
group-
very specific.
Yeah,
yes.
very specific. Well, what's your opinion in this kind of very large discrepancy?
Well, I've got some hypotheses that probably would be interesting to test, I guess. I definitely think it's a metabolism-based difference between salmonids. I think coho salmon definitely activate the quinone more rapidly than the other species do. I think there might also be some sensitivity in the mitochondria for as well in the target, perhaps. But I definitely think it's, if I go back to this, I definitely think the sensitivity is here for salmonids. Yeah.
All right, thanks.
Okay, any other question?
Oh, hey, co-author.
So much in your presentation, you said that some marine organisms have the detoxification ability to the natural toxins. So what's your opinion about the what are the real risk of these natural toxins to our ecosystem? Because I think they can detoxification by our marine fish or marine turtles. So what's the real risk of this natural protection?
Well, the author of the photograph of Kinney, Brian Brooks, actually would probably be very happy with that question because he does work on harmful algal blooms. And I think in terms of natural product toxins that are environmentally relevant and can decimate populations, I think that's a real good example. You see that happening, yeah, around the world. Mostly, I mean, climate change driven, nutrient inputs. You're seeing massive increases of those, and they're natural, totally natural products. And I mean, I would argue that they're just as damaging, if not more damaging than say legacy compounds that we're still dealing with, you know, in terms of population impacts.
Thank you. Thank
Yeah.
you so much.
Okay, great. Any other questions? Okay.
Oh, hello, Daniel. Thank you so much. I have two simple and silly questions. First one is about the how can we use this strategy, especially, you know, you did a very good job at a very deep level, for example, at the molecular level, and we use this deep level to understand how they survive in the severe environmental pollutions.
Mhm.
But how can we use this kind of deep and smart strategy to help other species to survive in the realistic environmental pollutions?
Mhm.
And another question is what is my curiosity. I really want to know how you feel to play a very important role in our community, because you play a very important role in our environmental science. You are very famous. Thank you so
Oh.
much.
Well, thank you for the kind words. Appreciate that. I'm not sure they're true, but I say your first question is how can this help us in terms of understanding risk, I think, of compounds. I'm a big proponent of if you understand how a compound works, you can understand the risks. I taught medical school for 3.5 years and pharmacology for almost 8 years. And the way that medicines are prescribed are based on mode of action. So if you can understand how a chemical is metabolized and how it is affected in different species, I think let's say in a conceptual model of a risk assessment, it gives you an idea of a species of concern. If you know that a salmonid, for example, is present in a location and there's going to be road runoff, you know, based upon what we know on the mode of action of these compounds, that salmonids are susceptible. So you would set your risk level at that species threshold, right? Because it's the most sensitive in that particular case. And again, you would know that based upon the mode of action and its ability to detoxify or not that particular chemical. So again, I think mode of action is the way, I think it's the only way of the world we will ever understand mixture interactions in the environment. And I'll go back, the average geriatric patient, you know, an old patient, takes 8 to 9 medications. That's a mixture. How do we know how 8 or 9 medications interact with one another? We do that by mode of action and looking at the mode of actions of those different chemicals to see if they overlap or cause toxicity or side effects between those particular cases. So again, I think the mode of action is definitely really important in understanding risks of compound. I think it's very important in understanding the weight of evidence that you put in risk assessments. And I think it's very important in identifying species of concern in conceptual models of risk assessments. So that's my 2 cents on, and I think it, you know, and the bottom line for any risk assessment is reducing uncertainty. And I think understanding how a chemical behaves in this paradigm reduces uncertainty. So, yeah.
Okay, thank you. Other questions? Okay, please.
As you mentioned, different butterfly fish have their own special detoxification strategies. So compared with generalists, the specialists maybe you know, under the global climate change or other marine pollutions, they have more risk of narrow ecologic niche.
Yeah, I think it's a good question. So one thing, I think the data actually bears that out. If you look at the hard coral specialist in Hawaii that only eats the hard corals, okay, and only occurs in Hawaii, doesn't occur in Australia, and we feed that compound to that fish, they die. Okay, but they're still eating hard corals, right? They're still surviving and eating really toxic stuff. But when something else comes off that they've never seen before, then they can't deal with that. So that population I was talking to Kenny about, the fungulus species on the East Coast, when they see hypoxia, they're much more prone to death than the animals that have never been in the polluted environment. Because they're putting all their resources into fitness costs associated with that one stressor, right? That's their one thing. And when something else comes along, change of salinity, change of oxidation, oxygen, dissolved oxygen levels, they don't have the ability energetically to deal with that, right? So you're putting all your eggs basically in one basket, feeding on that one coral, for example, which is still pretty amazing that you're able to survive to eat that. What happens is when something else comes along, like climate change or some new thing that comes around, then you can't deal with it. And again, that's why, again, genetic diversity is such an important aspect, is it allows you to deal with things that you don't expect, right? So that's why generalists will always outlive specialists, pretty much, unless that thing the specialist is doing overwhelms the generalist, right? That's the only case where that would ever happen, right? But in theory, generalists will always kind of survive any sort of modification in their environment, ecologically, so.
Okay, thank you. Now we have one online question from Lino Slow. She's from Education University of Hong Kong. And she said, thanks for the talk. And the question is, do we see any other sponge grazers capable of similar detoxification while storage pathway?
Do we see, I'm sorry, do we see any other,
Do we see any other sponge grazers?
oh, sponge grazers?
Yes.
Yeah, so, not that I know of. I mean, there are sponge feeders. There's another fish species, the name escapes me right now, that does eat sponges. Oh, emperor angelfish. Emperor angelfish. This is one thing we wanted to look at, actually. Any students out there want to look at emperor angelfish? That's a good one. It's a sponge eater. And I would love to look to see what its GST levels are like. I bet they're screaming very high. I mean, I would hypothesize that. But, and they're interesting because they changed their feeding strategy based on juveniles versus adults in terms of eating on the sponges. So, but I know that fish species for sure eats sponges.
Okay, thank you. So maybe we have 5 minutes left, and maybe we can have the last one question if you have. Okay, if no any other question, then maybe we can talk more after the lecture.